Hello from Chem Help ASAP. Let's outline some of the later preclinical safety studies for a drug program. By the end of this video, students will be able to describe preclinical studies that are often performed after clinical trials have already started, and justify why some preclinical and clinical studies can be performed concurrently. Here is a slide that shows drug discovery and many of the preclinical safety studies that are performed during drug discovery. These studies provide evidence of the safety of a compound and enable the initiation of short-term safety studies for acute toxicity in Phase 1 trials. If the compound advances into Phase 2 studies, however, additional preclinical toxicology studies must be completed to support prolonged use of the drug and the inclusion of a broader range of patients in the trials. Let's discuss these preclinical studies that support Phase 2 and Phase 3 clinical trials. The length of GLP toxicology studies depends on the length of the expected drug treatment. Drugs with a short dosing regimen, something like an antibiotic, may only be dosed for 2 weeks or less, and the toxicology studies may only be conducted for 1 month. As the length of treatment increases, the length of toxicology study also increases. Note that the planned length of treatment can change as a compound advances through clinical trials. If the anticipated length of treatment is extended, then additional toxicology studies may be required. These studies emphasize the general health of the animal, although specific risks may be monitored based on the mechanism of action of the compound. This slide covers studies that monitor different aspects of reproductive and developmental toxicology. Fertility studies treat animals, normally mice or rats, both male and female, before and after mating and up to embryo implantation. The fertility of the animals is monitored. Embryo-fetal development studies involve treating pregnant animals with the drug through closure of the hard palate. Examination of the fetuses looks for any physical abnormalities. Pre- and postnatal development monitors the offspring of treated female animals for viability, growth, and development. These studies continue through sexual development and may include a mating phase. Collectively, these reproductive and developmental studies help inform women of childbearing potential of the risks of using a drug during pregnancy. Long-term carcinogenicity studies are required for drugs that will be taken for extended periods. The animals in the study, either mice or rats, are dosed for up to 2 years, which is very close to the expected lifespan of a mouse. It is possible to use transgenic mice, which can shorten the study to 6 months. During the study, animals are dosed with the drug, and tumor development and progression are monitored. Drugs that are being developed for oncology indications may not require carcinogenicity studies unless patient life expectancy is expected to be 5 years or longer. We have now described many of the common preclinical safety studies: long-term toxicology, reproductive and developmental, and carcinogenicity studies that are performed after clinical trials have started. We also justified why some preclinical studies can be performed concurrently with clinical studies. I hope you enjoyed this video and found it informative. Please consider leaving a like, making a comment, asking a question, or subscribing to the channel. Take care.